Hello everybody, my name is JRodriguez. I'm a senior at WM High School, and I did my senior internship on detecting protein misfolded states. And that may sound very confusing, but it really isn't. Basically, we're trying to identify a protein that has been altered or changed by some external influence, such as a virus. So proteins are the building blocks of life. Without proteins, biology as we know it would not exist. We wouldn't be able to stand, we'd just be pooping on the floor. So proteins have a variety of different tasks and functions around the body. Proteins have a variety of tasks around the body. They can help prevent the over reproduction of cells in the body. However, proteins are easily susceptible to change or misfold in this case. The geometry of a protein is everything when it comes to a protein's performance and its ability to perform its action. So as you can see here, this protein right here is the native state or the normal protein. And let's say you have a protein shaped like a triangle or shaped like a square, and a virus comes along. That virus can easily deform that protein into a triangle, and therefore that protein will no longer work properly. So to start off my research, I had to think of the question, is it possible to cure viruses inside the human body through how a protein misfolds? And basically there are two forms of a protein that I mainly looked at, the native state of a protein, and then also the misfolded form of what changes with the protein after it's been misfolded by some sort of virus or other external force. My internship was served at the Virginia Commonwealth University in the Chemical and Life Science Engineering building. And basically, VCU started up this program actually this year. This is a new program, and it takes rising high school seniors into a college research environment. The students are paired with a graduate student as their mentor. They work together to solve real world problems and attempt based on whatever project they're doing. My mentors for my internship were graduate student Oscar Bastidas and Dr. Michael Peters. Oscar and I, we worked in Dr. Michael Peters' lab pretty much my entire internship. Oscar is he recently received his master's degree in chemical engineering, and now he's focused on getting his PhD in chemical engineering. And during my internship, I had the opportunity to interview Dr. Peters. I was really amazed of how he got his PhD in chemical engineering, but his interests were in more biological sciences with proteins. During my interview, he told me that whatever research you want to do, it will follow your interests. And I just thought that was amazing, because even though you are focused on one field, you can still branch out and see what other things you can do. Oscar and I, during our research, we did a lot of computational data, which involves looking at the native state of a protein and the mutant or misfolded form. So basically, we looked at the common amino acid interactions between both proteins. The amino acids are what make up proteins, and the common amino acids in both the normal native state of the protein and in its folded form, we were able to find a computational solution to see which amino acids did not change from the virus misfolding the protein. And this set of amino acids is a very important factor, because with this information, we would be able to figure out how to potentially genetically engineer another protein that can refold the protein back into its native state. And so this topic of proteins needs to be researched a lot more. The sky of proteins. Going into this, I knew what a protein was, what it can do in the cell, but I had no idea that it had all these functions, like what exactly they do. Like, there are so many different proteins that help protect us from several diseases, such as ALS, HIV, and even Alzheimer's. And a virus can just easily misfold that protein. And let's say that protein was to stop the reproduction of cells. If that protein was misfolded, then there's no protein, nothing that will stop cells to continually reproduce. That would be an over reproduction of cells in the body. And so some possible solutions for this problem is right here. This is a prion. Prions are well-known diseases in the brain, like mad cow. And basically, with the knowledge of what amino acids are being used inside of the virus and also the protein, with those amino acids in mind, we can genetically engineer this prion to target whatever the virus is searching for. If the virus is searching for a specific protein, the prion will target, search and destroy by pretty much doing the same thing what a virus does to a protein. It will misfold its own proteins, making the virus useless. And then also, this is a dialysis machine. People with diabetes need to undergo dialysis very frequently to clean out their blood. And so Oscar and I came to an amazing discovery that viruses, they need to attach onto the healthy cell to be able to inject its DNA to misfold the protein. And so they have a lock and key mechanism where the viruses have all the keys to all the locks of a cell. So we thought, if we insert a microfiber into the dialysis machine when the blood is going through the machine, the microfiber would contain similar receptor proteins to the virus itself, so that the virus will think, oh, these receptor proteins are what I'm looking for. I will attach onto them. And basically, they will stick on there and clean blood will flow through. And basically, the viruses can be drowned in bleach and kill off those viruses that were in the host body. My community service, I volunteered at Thrive Healthcare. They're a local nonprofit organization here in Charlottesville. They deal with the treatment and conditioning of people who have AIDS. They are really passionate about helping people, calming them down, because people who test HIV positive think that their life is over. I mean, you have a disease that is not curable, and they think their life is over. There's nothing they can really do. And Thrive pursues to convince and help people who have HIV to there are ways to treat HIV, to limit the number of HIV cells inside a human body. For my mentors for my community service is Caitlin Wernsing and Eric Mays. Well, my time at Thrive, I basically helped file paperwork for patients, send out thank you letters to donators, because since it is a local nonprofit organization, they have had problems in the past with budgeting. And and also help set up whatever volunteering events they like to do. Eric Mays, he helped me with a more internal volunteering and drive. I was able to shadow a group meeting where public people who are HIV positive get together. They talk about how they're doing their lives. When they first found out they had HIV, just how tough they had it when they first found out. Some of the stories were really amazing, or amazing how they got through it. Some of them, as soon as they found out, like I said, they thought their life was over. They went through drugs. They were homeless for a lot of time because they thought they had nothing else to live for. But it was amazing. Sitting down there in that meeting, they have a great time. They share dinner together and they just talk about how they're doing and what they hope to do in the future. Another part of my community service, I helped out the annual 5K AIDS awareness run walk. Basically, I helped set up all the festivities that were going on. And see here all these balloons in the downtown Charlottesville area. I also ran in that race to help raise awareness, and I felt like I was helping out Thrive by raising awareness. This 1.2 million right here, that is the number of people who have HIV in the world. And about 7% of that are people who are unaware that they have HIV. That's why Thrive urges that people come in to get a checkup to see if they have HIV and give them opportunities of what they can do in the future to limit passing on HIV to others and also limit the number of HIV cells inside of them. My legacy, during my time, I have been able to publish 2 papers as a co-author, along with the help of my mentor, Oscar. One of them right now is published actually, and the other one is in peer review. So I was really excited that I could be a part of an actual paper that is submitted. And also for my internship, I did a total of 162 hours of dedicating my time to research how we can stop these viruses from exploding proteins. And I thought it was just an amazing experience for me. Self-reflection. At first going into DRE, I thought, okay, I'm just going to go in the lab. There's a bunch of biosafety modules that you have to go through to train so that you are prepared if there is a case of contamination or anything. And also, Oscar himself, when he first started, he was during his undergraduate at VCU. He was able to mentor by Dr. Peters. That's why he started to become very interested in proteins. And he's told me like all these experiences he's had during college, and he was really amazed at what he could do with the help of Dr. Peters. And things that I would do differently, I wish I could have done more with my community service, like maybe have a AIDS awareness day at the high school or something to just raise more awareness even around Charlottesville or Greene County. Advice to my future senior members, future Brewery seniors. Do not procrastinate. The deadlines where they just jump on you like that. I mean, senior year is going to be very busy, college, and you're just gonna have all these deadlines. Try and get an internship or community service over the summer. That just really helps me out because most of the time during the summer I was training to go into labs, and research really, and then finally have fun with it. You're going to do this project for the entire year. And my future plans, I plan on attending PVCC and then transferring to Virginia Tech, and one day continue my research with just as Oscar did, and hopefully find a cure, a universal cure to all viruses. Any questions?
So this discovery with the dialysis process, I mean, what you guys were trying there, you touched on it just a little bit. Can you explain a little bit more about kind of the outcomes of that? So you're putting in, just can you explain that process? And also, has it gone on, as there continued research on it?
The only research we were able to do was to figure out from a list of multiple proteins and what their functions are. We didn't do any testing with the dialysis machine. This was more of an idea, like with the receptor proteins on the cell that would be placed on the microfiber. Those viruses, at first we thought that this microfiber wouldn't have much of a effect, but we realized with the addition of the receptor proteins, it will actually has a lot of significant help. Does that answer your question?
It does. Were you doing lab tests on that? Were you doing computer modeling? How are you?
It was mainly a bunch of Excel spreadsheets, comparing the amino acids to the form of the protein.
Okay.
And basically, I looked at the different interactions between those amino acids, see their distances, and see if they actually stay if the protein is misfolded.
My math is correct based on your data. There are 85,000 people walking around with HIV who don't know it. What are we doing to find those people and help?
Well, one, raising awareness really helps, especially if your family has a history of HIV, because my mentor, Eric Mays, he actually has HIV. He has not told his parents but he's been on newspapers and talk shows about him. And I really think raising awareness to even people who are unaware what HIV is or if they potentially had it would really help make their mind curious to see what they can do for that.
Just curious in terms of whether in your research you did anything about awareness and spread in other countries. You know, that there's Africa, it's a huge problem, and growing one in Asia.
Sorry, can you say that again?
Just whether you came across your research about what's going on in other parts of the world, AIDS prevention, and
in my research paper, I put in a section about the Ebola virus that was in Africa, and I found out that this technique of either using prions or looking for those amino acids of what proteins Ebola misfolds for it to cause such a pandemic or epidemic. But other than that, just the Ebola virus that I really looked into great detail.
What part of the research lab did you enjoy the most? What parts of the research field that you think you're going to go into in the future?
Working on a research team and definitely using a bunch of the laboratory equipment. Like Oscar and I, we one day we found like a bunch of insects, like just roaming around the lab, and I'm like, ooh, and they're already dead. Like, ooh, I wonder what happened if we look at those under a microscope, and just looking, and it's really cool messing with like the centrifuge, a fume hood, using just different laboratory equipment. On the research team, I think it's really interesting because Oscar, Dr. Peters is his official minutes work, and I find it really interesting. Like, you really have to proactively communicate with your research team. Like, you always have to ask the next set of questions to your advisor and to all the other members of your research team. Another person on the research team was a student. He helped us a lot with the computations, and working with him was a great help, actually. And I just thought it was really interesting. A variety of professors, students,
So you had fun in those labs?
a lot.
Oh,
A lot.
okay.
It was great.
Thank you, Jeff.